Hello, welcome back. This is Professor Young, and we are continuing our journey with the urinary system. This is part two of three Limbus sessions that will be devoted to looking at the renal system. We left off, I believe, on the last Limbus session, talking about the beginning structures of a nephron, and that would be the glomerulus. Which, of course, we know has an afferent and an efferent arterial that is going to deliver blood into the glomerulus, at which point in time the filtrate any kind of waste products, excess waters, things of that sort, will then enter into that Bowman's capsule. And from there, it will lead into where we are talking about today. So just kind of pointing out, so far in our journey, this is what we have looked at so far. And what we now need to begin to look at is what happens to this filtrate once it has been removed from the cardiovascular system, from the circulatory system, and where exactly does it go after it has been absorbed and put into Bowman's capsule. And to answer that, we have four very distinct regions. Three of these are going to be associated with the nephron, and one is not. But just kind of getting us oriented here once again, we have region one, which is the proximal convoluted tubule, which will lead into the loop of Henle. And then from the loop of Henle, we've got the distal tubule. And then we have the collecting duct. If you're looking for a rough beginning and end to where everything, begins and ends, of course, where this blue line is the approximate area of the beginning part for the proximal tubule, and that proximal tubule is going to end approximately within this area right here. From there, we enter into the loop of Henle, which would continue down and around to approximately this area right here. And then, of course, your distal tubule would basically be the rest. And so your distal tubule would start right about there, and it would end right here, and then the remainder of the space right here would become your collecting duct. And so they're kind of the areas that we have outlined for us that we need to pay attention to. The other thing I do want to mention is if we are dealing with a cortical nephron, then everything, with the exception of approximately where this black line is down, this is the area that is going to be found within the medulla. The rest of this structure is going to be found in the cortex. Whereas if we were dealing with a juxtamedullary nephron, we would see a lot more of the structure. Let me erase that. We would see a lot more of this structure found within the medulla, basically from that black line once again all the way down would be found within the medulla. And the rest of it there above would be found within the cortex. And so kind of keep that in mind as we go forth and look at this. And in fact, if we pay attention to, and I'll just point this out real quick, right down here, we have that image of a renal pyramid, and you can see even further down. You can take a look right into this blue area. You see that the majority of the loop of Henle is in the medulla, and so even here they're showing us basically a juxtamedullary nephron. So just kind of keep that in mind when we talk images. You're typically going to see that juxtamedullary, but in reality, majority in humans of the nephrons are going to be the cortical nephrons. All right, so why don't we go through and we begin our journey looking at each of these different structures and aspects of the nephron. We know that the beginning section is referred to as the proximal convoluted tubule. It is outlined there in red for you. Simple cuboidal epithelial, mainly for storage purposes, and we're gonna talk about this a little bit more. Lots and lots and lots of microvilli. The microvilli is there for the purpose of increasing surface area. And again, the function here is to absorb. The glomerulus is going to filter, and then we're going to see some reabsorption happen here, which is why those cuboidals are so important, because again, it's going to provide for extra storage while we are reabsorbing. And it is highly convoluted. One of the reasons, speculatively, why we have a highly convoluted proximal tubule is to increase the length within a very short period of space. And so we have high surface area with the microvilli. We can, by folding it as much as we do, we can increase the amount of length of the tube that we have within a very small space, and all of this allows for more time and a greater ability to reabsorb. Going from the proximal tubule, we enter into the loop of Henle. And if we were to give it its own color, again, we are talking about depending on the nephron, but we're talking about approximately that area in blue. It has that very characteristic U shape. Notice the narrowing down at the base of the loop of Henle that occurs, and then again it widens back up. The thicker areas of the loop of Henle is simple cuboidal, whereas the thinner areas are simple squamous. Part of that has to do with the pressure gradients. As the loop of Henle descends down into the medulla, the pressure increases. And if that pressure increases, now think about this, because this goes back to that whole idea of pressure gradients. If the pressure outside of the loop is greater than inside of the loop, are we going to be able to have any kind of reabsorption occur? Yeah, then probably not, because we know that pressure is going to move from an area of higher pressure to an area of lower pressure. And so by narrowing that loop of Henle down at the base that's being surrounded by higher pressure, you increase the pressure within the loop. And what that does is it allows for a more even flow of particles. And we're going to talk more about that. But by design, this does make sense. And again, that loop of Henle does extend down into the medulla for both types of nephrons, both the cortical nephrons and the juxtamedullary nephrons. The distal tubule is where we see, kind of do an outline here for you. distal tubule is going to be approximately within that blue area. Again, this is an area that is found within the cortex. So we start in the cortex. The glomerulus is in the cortex. The proximal tubule is in the cortex. The majority of the loop of Henle is in the medulla, and then we enter back into the cortex with the distal tubule. Cuboidal epithelials doesn't really need to have the microvilli because we're not really doing any kind of reabsorption or filtering or anything like that. And it does signal the end of the nephron. It does signal the end of the nephron. So that's the down and dirty overview of this structure of the nephron. And of course, we have one last little aspect that we need to mention, and that being the collecting duct or the collecting tubule, which is right here in purple. And what that is going to do is it's going to drain the fluid from several nephrons. And so the fluid that is moving through the distal tubules right there will enter into the collecting duct. And from the collecting duct, it will go into the papillary duct, which is going to be found at the base of the pyramid, right down here. So that renal papillary or papilla, and then that's going to extend down into the minor calyx, which is going to go into the major calyx, which enters into the renal pelvis, which will then eventually empty into the ureter. So, how does all of this equal urine? Well, we have three basic processes that occur, and those three basic processes are outlined right here. We've got filtration, we've got reabsorption and secretion, which we will talk about, and then we've got excretion, which we're not going to spend as much time talking about. And basically, the picture that you're looking at here to the right is a oversimplified version of what is happening during this whole process. But you can see that during reabsorption, we remove any kind of useful particles from the filtrate that was removed within the glomerulus, and that material is returned back to the blood. And then during secretion, we remove again any kind of waste products that are happening within the blood, and we move it back into the filtrate. And so up in this area here, we've got, again, filtering, and then from filtering we move into reabsorption, where we move from the filtrate back into the blood, and then from there, again, we move during secretion. They're secreting from the blood back into the filtrate. And then later on, water is removed from the urine because water is a beneficial aspect of it. And then what is left over at the very base is the urine, and that is what is gotten rid of. And so you can kind of see this progression. You can see this progression of this back and forth aspect, which will make perfect sense to you by the time we are done with this. We are concerned as of right now about filtration. In fact, the rest of the slides that we're going to be looking at here are going to be focusing specifically on filtration. And so when we talk about filtration, we're talking about that movement of fluid out of the capillaries being surrounded by the glomerulus into the capsular space, which of course we know that the technical name for that capsular space is Bowman's capsule. And really what is happening here is we've got a series of 3 steps or 3 layers that filtrate needs to that filtrate needs to pass through in order to ensure that only the essential waste products are removed. And when I say essential waste products, what I'm talking about is we want to make sure that red blood cells are not being removed. We want to make sure that proteins are not being removed. We want to make sure that only things that are in excess, such as water, or things that are waste, such as the nitrogenous waste, are being removed. And so we have these 3 membranes, or these 3 layers: the fenestrated capillaries, the basement membranes, excuse me, and the filtration slits, that are going to regulate this process. And so here's what these 3 layers all look like. The first layer, again, that we have is right here, which is pointed by the arrows. And again, that would be the fenestrated capillaries. And that's going to be the first line of defense, if you would. From there, we move into the basement membrane, which is right here. And then lastly, we've got right up here, and by that top arrow, we have what is amounts to the filtration slits. The filtration slits right there. And so what ends up happening is the blood is coming down. Anything that is considered to be a waste product is going to pass by those fenestrated capillaries. If it is definitely something that is that can pass through the membrane, and basically any kind of small particle or anything that is positively charged will be allowed to pass through the membrane. And then if it's still small enough, then it will be able to get past the fenestrated slits. And so that way it will go ahead and remove it. Things that will be passed through the filter. You can see a whole list of things there: water, electrolytes, glucose, amino acids, fatty acids, vitamins, urea, uric acid, and creatine. Specifically, we have in the past already discussed those bottom 3. We know that they are all specific types of nitrogenous wastes. Water should not be a biggie. And a lot of the substances that you see listed right in right in this area, the electrolytes, the glucose, the amino acids, the fatty acids, and the vitamins, eventually will all become reabsorbed. One of the telltale signs of a diabetic is when glucose is found or present within the urine, and so that is one of the things that are definitely going to become reabsorbed. And of course, water we know is reabsorbed as well, and that's a hit or miss thing depending on how hydrated you are. Now, one of the things that you should be mindful of is that if the molecule is too large, and specifically I'm looking right in this area here, if the molecule is too large, you can see that it will basically be rejected or rebounded. It won't be allowed to pass through those fenestrated capillaries. And it will never get into the membrane, which means it's not going to pass through the filtration slits. And so that's a built-in process. And notice how selective things become here. The fenestrated capillaries are a little bit larger. You get a little bit more stricter of a standard when we get into the basement membrane. And then, by the time you get into the filtration slits, look how regulatory they are. And basically, these podocytes, these foot-like projections that you're looking at there in brown, can change shapes and change widths and diameters, which means it can regulate just how selective it wants to be in that specific case. All right, hydrostatic pressure. We know that we can, or at least we have discussed within lecture, the fact that by manipulating the afferent arterial and the efferent arterial, we can go ahead and manipulate the rate of filtration as well as blood volume and blood pressure. And just so that we're all on the same page, we should hopefully be able to identify this as the afferent arterial, and then this guy down here as the efferent arterial. And so blood is entering in through the afferent, it is filtrating itself, and then it is leaving through the efferent, just in case you weren't following those arrows. But basically what happens is, and just as it says on the slide, high pressure due to the large afferent in, small efferent out. And so you have a high amount of pressure found within the glomerulus. That's where the high pressure is. And that high pressure is going to come from the fact that the efferent arterial going out is a smaller diameter. Smaller diameter means that it's not going to be pumping out as fast as the efferent is bringing in. So your blood flow coming in is much greater. The blood flow going out is much less. That causes a backup within the glomerulus, which is going to raise the blood pressure. Right? Or not necessarily the blood pressure, but the hydrostatic pressure found within that glomerulus. All right. Now, within the capsular space, we're also going to see a high rate of filtration. Why are you going to see a high rate of filtration? Because the hydrostatic pressure, the pressure of the fluid pushing against the glomerulus, is higher than what it is within the capsular space. And we know that pressure is going to move from an area of higher pressure to an area of lower pressure. All right, so where you see in the yellow arrow, BHP, blood hydrostatic pressure. Blood hydrostatic pressure is 60. Now that's 60 millimeters of mercury, don't forget. I'm just going to start leaving off millimeters of mercury. I think I beat that into your heads enough throughout the semester. So the blood hydrostatic pressure is 60. The capsular pressure is only 18, I'm sorry. It's only 18. 60 pressure pushing against the glomerulus. 18 pressure within the capsule. High rate of filtration. High rate of filtrate moving from within the glomerulus into that capsular space. All right. And throughout this whole process, as long as you are in the glomerulus, the blood hydrostatic pressure remains high. It has to remain high because otherwise you run the risk of causing reabsorption. And if you have reabsorption within the glomerulus, you're putting back into the blood supply all the nasties that we just got rid of. And so we have to allow for a consistently high blood hydrostatic pressure within that glomerulus. That just has to happen. Has to happen. And so hopefully that makes sense there when we talk about that beginning section. Now, how fast does this all occur? And how much are we really referring to? Well, in the typical scenario, and by the way, where you see, I just want to make sure that you're understanding my hieroglyphics here. Actually, it's my laziness. NFP. NFP simply stands for net filtration rate. That's what NFP is standing for. And so when we talk about filtration within the glomerulus, we're talking about how much of that filtrate is being produced per minute by both kidneys. So this is both kidneys working with one another. All right. Typically, that net filtration rate is going to be about 12.5 milliliters. Right? It's going to be about 12.5 milliliters. And so for every minute that goes by, your kidneys are filtering 12.5 milliliters of filtrate. That's a high amount of filtrate. Right? Now, keep in mind, that's not necessarily how much urine we are producing, because we know that there's going to be a lot of reabsorption that's going to be happening, and we'll look at that again in lecture. But basically, you're talking about 12.5 milliliters of filtrate. Now, some of this there's some variations in this. Permeability. We just got done talking about those filtration slits, and we know that they can become more selectively permeable, or they can decrease the amount of selectivity that they have. And then again, the surface area that we're dealing with as well. And that all goes back to those three layers found within the glomerulus. Pardon me 1 second. Okay, sorry about that. So, what exactly is regulating this 12.5 milliliters of filtrate that is being produced every minute by both kidneys? Well, what it comes down to is really a negative feedback system. You have the tubular glomerular feedback. The glomerulus is going to be regulated or controlled by a structure known as the Juxtaglomerular Apparatus. And basically that is located at the end of the loop where it touches the afferent and the efferent vessels within the renal cortex. And I've got a picture of that on the next slide, so I'll be able to point that out a little bit more to you. But that is what this is. That is what's regulating everything. In fact, let's go to it right now. And so, if we look, the efferent arterial is located right here. The afferent arterial is located right here, and you can see those juxtaglomerular cells are located right where that arrow is. And basically what that is doing is it is regulating that fluid. It's regulating pressure changes. So that way it will allow for maximum filtration possibilities within the glomerulus. And so it's not measuring it on the way out, it's measuring it on the way in. It wants to know how much pressure is coming in. And based on how much pressure is coming in, we can regulate the afferent arterial, whether it needs to constrict a little bit more, whether it needs to widen and open up a little bit more, or whether it needs to go to the efferent arterial and say, hey, wait a minute, we need to do something with you. The juxtaglomerular apparatus or the juxtaglomerular cells is what's going to regulate that. And so that's going to regulate, again, that's going to regulate that 12.5. That's going to help to regulate that 12.5 milliliter per minute filtration figure that we just looked at on the previous page. Now, not quite done with us. Almost. All right. We can't escape the system without at least somewhat talking about the sympathetic nervous system. And basically what happens here is those renal blood vessels take cues from the sympathetic nervous system. It has to. All right? It has to. Because as we know, the sympathetic nervous system is going to be functioning in high gear during extreme or during any kind of physical activity. And so you're running, you're walking, you're jumping, you're jogging, you're hopping, whatever it is. You're yelling at someone. You're screaming at the top of your voice, and you're ready to shoot someone, or or whatever it is. The sympathetic nervous system is going to play a role in that. And so what the sympathetic nervous system does is basically restricts the amount of filtering that is happening. And think about why would we want to do that? If you're restricting or if you're constricting the afferent arteriole going into the glomerulus, you're reducing the amount of blood that is available for filtering, but you're also redirecting that blood into the skeletal muscle. And so that is important. And you can see the blood is redirected to the heart, the brain, and the overall the skeletal muscle. When you do that, you reduce the GFR. All right, now, for those of you who don't quite remember, GFR simply stands for can't spell glomerular filtration rate. So exactly that I still messed up the spelling on that. So exactly how much filtering is happening within the glomerulus is what we are looking at, and that again goes back to that 12.5 milliliters. So when the sympathetic nervous system is kicked into high gear and is responding to you running from your car into class because you are running late, what's going to happen is your rate of filtration is going to go down within the glomerulus, and that's going to redirect that blood flow to the skeletal muscles. Now, there's one last little thing that I do want to touch on, and then we're going to end here today. There is this complex system known as the Renin-Angiotensin-Aldosterone mechanism. You see a nice little diagram in front of you, and basically what this is going to do is cause vasoconstriction. It's going to cause the vasoconstriction to help raise or elevate blood pressure. Now, what happens is this whole system is activated when BP drops. And so when your blood pressure drops, and typically this is really going to be activated if you are bleeding out, and that's the cause of the drop in blood pressure. But for argument's sake, we're just going to say whenever blood pressure drops and drops severely, not just a little, but severely, we know that renin is released by the kidneys. We talked about that a little bit earlier. And so what's going to cause that is the sympathetic nervous system. It's going to cause the juxtaglomerular cells to release the renin. Angiotensinogen is released from within the liver. When renin comes in contact with the angiotensin, or I should say when renin comes into contact with the angiotensinogen, it cleaves it and forms angiotensin 1. Angiotensin 1. And then from there, the lungs are releasing an enzyme that is then going to cleave it into angiotensin 2, you following this? Yeah, blood pressure drops. Renin is released from the juxtaglomerular cells within the kidneys. It's going to cleave or change angiotensinogen, which is being released from the liver. It's a protein. It's a protein, and it will then go through and cleave the angiotensinogen into angiotensin 1, which will be further cleaved into angiotensin 2. Angiotensin 2 will stimulate the hypothalamus within the brain to trigger thirst, which will have you drink. It will vasoconstrict blood vessels throughout the body, and then it will also act on the kidney itself through aldosterone, which is a hormone, and that will cause vasoconstriction within the afferent arterioles, which will again help to retain salt as well as water, and the combination of all three of them will help to raise blood pressure. And so on that note, I will leave you to ponder, think, and review. And as always, if you have any questions, please feel free to contact me.